Let me share my screen. Full screen mode. Yes. So I want to discuss with you I want to introduce you to a tool which is based on deep learning, which is called AlphaFold, which is a deep learning tool to predict the structures of proteins. And the reason why why we use this tool is because proteins perform all sort of functions in nature. So proteins are not just what you are used to see, are not what you are used to see like you imagine like in the meat or milk, but every function in your body is driven by a protein, like from the color of your skin or your eye, or if you are predisposed to a certain type of treatment from a specific medicine. And this has been is highly relevant, especially now that you are seeing that we need very quickly to find a vaccine and cures for some disease. Why I am talking here about this is because there is a well-known competition where usually expert of biochemistry participates, and these are like top groups of working on biochemistry. Some of them even affiliated to big pharmaceutical company like Pfizer, Novartis. And what has happened is at the last of this challenge, where people were competing on how to predict structures of proteins, there was also a group from Google called DeepMind who participate. And what happened is that they outperform these people from this biochemistry lab. So these DeepMind people were people that were used to make before recommendation, like for YouTube or self-driving cars. They were not expert in biochemistry or people with a medicine background. Probably in the team they had someone who was knowledgeable of biochemistry, but substantially they were a bunch of nerds, so software programmer. So what happened is this software programmer, the tools that they developed was even better than these professional people from university, and this made up in the news, so was a bit shocking. So what they did is to apply like advanced deep learning techniques to reconstruct the protein structure. Now, if you remember, like some deep learning techniques are substantially there is a parallelism with our visual system. Like imagine, like for the visual system, you have you are observing an object. So there is a radiation hitting the light radiation hitting the object, and then bouncing back to your eyes. And then in your eyes, like from this is translated to some impulse in the optic nerves, going somewhere in a part of the thalamus called the lateral geniculate nucleus, and then irradiating here to V1 in occipital lobe, and then going, if you consider only the dorsal pathway, is going to other series of layers of neurons. Now, we can see some deep learning approach that kind of reproducing this path. Namely, of course, you don't have the real eye or the lateral geniculate nucleus, but you have a picture, which, so the objective of a camera is like reproducing the eye. So you are acquiring this, and then with convolutional network, you do the work that the V1 area is doing, because it's this area is responsive to some edges. And then you have a series of computational layers which are similar to what we have in the dorsal pathway. You can look up this paper to have further discussions on this parallelism. But the idea is that of DeepMind was that why we should consider only deep learning architecture for vision, we should also use it for other kind of problems. More specifically, what they did is instead of moving from picture to some kind of semantic representation, they moved from a series of genes, a series of pattern of amino acid to a protein structure. So this was the problem they tried to address with this tool called AlphaFold. So that I'm representing here, so is that what I was telling you. So from a sequence of amino acid, if you can predict the structure of a protein, that's it. So we can close it here. You can go to lunch. If you got this, no, okay, no, unfortunately, no. We should go a bit deeper in how these things work. So because so I go super quickly. So this is less than biology 101. Probably if there is someone with among you with a background in mathematical biology or bioinformatics, this will be an oversimplification. So I apologize also for the terms I'm going to use, but I'm just to boil it down to the people who are not familiar to this. So the idea is that we, from proteins, have a primary structure, just a sequence of amino acid, and then we have a more advanced structure, in particular the quaternary structure, where they fold in a specific way. And this is important because how they fold influence many things. So even certain diseases are caused by proteins that are misfolded, or as I was telling you, depending on how the way the structure they interact, that's how you can see the reaction for a specific treatment with specific medicines. I'm going to go super fast, summarizing the story where these things are from. So in your body is made of cells. So in the cells, you have a nucleus. Inside the nucleus, you have the chromosomes. And chromosomes are made of DNA. They hold the DNA, and the DNA is made up of genes. Another further subdivision is that the DNA is not just DNA, but it can be transcribed in RNA, which is like the single string, and then translated into a series of proteins. Now, these translations, you have like a precise order to which the amino acids are made, are collected one after the other, because every protein has a unique sequence of this. So this kind of translation is also it's what was happening to build the initial structure of the proteins. Now, to obtain what I was telling you, to obtain, ended up in the all the other structures, including the quaternary structure. There is something this is happening in nature by itself. So they fold according more driven by physics than the law of physics. Namely, that the structure that they will obtain is in such a way that that require less energy. So you can see if in this graph I'm putting here that the real native shape of this structure of these proteins will be the one that require less energy. This is not, this is of course the results of evolution and of the laws of physics that we cannot easily predict. Because if you want to imagine these things, it will be combined in all the possible combinations of how these atoms are how these amino acids are connected, it's too complicated for us to predict. There are some techniques that we use that I mentioned here, like cryo electron microscopy, nuclear magnetic resonance, X-ray crystallography, that they are able to reconstruct the structure, but those are highly complicated, and also sometimes they can deteriorate the samples you're looking at. So it's not the very easy way to reconstruct the structure of a protein. So what has happened is instead is that the sequencing has been cheap a long time. Sequencing is the way that we have to know the particular sequence of DNA in something. Is becoming cheaper, and this means for us that there is also some of these data sets are online, and it's also a good thing that they're online because it means a lot of data that we can use to train algorithm. Even the new COVID-19 has been reconstructed, and the reason why this has been reconstructed is because it has some spike specific. I can send you this paper later where they've reconstructed the whole structure. So this one I'm showing before was just the main structure, but they've like spikes that are these red things that you probably have seen in many pictures. I'm looking for a I don't have a picture, but I guess you have seen in this structure where there is like a sphere and some spikes. So these are specific receptors that they attach to the human cells. So if you can design better proteins that interact better with the in a way that inhibits the spikes, so instead of going to human body, you block them. You can design like vaccine or treatment for coronavirus also. So it's not just, and what I'm telling you is not just in the air. Like there are people really focusing on these things. In fact, I can send you the link. Actually, I put it in the. There is this group Foldit. If you can find, there is a simple video where they are explaining how they want to block this spike with receptors. Anyway, we are not here, at least not to solve it today, but I'm here to show you how this technique AlphaFold is used, and maybe I don't know, after lunch you solve you use AlphaFold and for finding a cure for corona. Okay, so where was I? Yes, that we are okay. Too much. That's it's going too much. So I'm telling you that this we have a lot of genetic information, and our idea is that we will use we can use all this information. So in practice, how AlphaFold works, I was telling you, you give a sequence of amino acid and it's predicting the shape, the structure. Now, this is like also related to another observation, you see that I told you that the structure is given by a conformation that is minimizing some energy. These things looks familiar to you? Have you already seen this?
Yeah, hello.
Yeah,
so I'm saying that you can also look at it from the optimization point of view. So as you explained from the previous class with the curve, kind of using the gradients, sometimes taking the small steps, I mean, you would be able to get the minimum value for the energy.
Yes. So, okay, I mute you. So what they said is correct. So what is happening in nature is that they are converging to a structure with using the less energy as possible. And what is similar to what we do with the gradient descent computationally, we find a minimal error. So there is like a parallelism with what we can obtain with computational models and what nature is doing. It's here. So this is, you don't see probably because of this. So you see it's in like, this is like kind of natural behavior. Is in the end the protein is folding, tend to fold to this conformation where the energy is minimized. This is not our gradient descent. This is the nature, like how in reality a protein is going to fold. So there is Deo. Yeah. Deo, you're on. You're up.
Can you hear me?
Yeah.
I wanted to ask, you talked about the protein structuring and all that. Is it in relation to sequence alignment of DNA and the RNA structures?
Yes, the alignment is multiple alignment is actually the initial step in that this technique used to find similarities with other structure. But then there are also other things within the system. So okay, since now you're probably curious to know what they are doing. So the idea is, as I was telling, in a similar way that in a similar way that people were doing for reproducing the visual system, so that they train specific features so that they are connected to a specific semantic value, like there is a cat in the picture, or there is Marie Curie in the picture, or there is a face in the picture. So you link the input is the picture, and the output is there a cat or a dog or a face. Here they give sequence of amino acid, and the output is the structure, which is okay. Before I go, with structure, it means that we are supposed to know the coordinate in space of the singular atoms building up the backbone of the protein, and also what is the angle in this kind of bonds. So how are these angles organized overall? So I have a video to show you, not made by me, but from Mohamed Al-Kirash, where I try to turn back, where I'm showing you better this. So this is like, imagine this is the backbone of the protein, and we have, of course, nitrogen, carbon atoms. What we are interested is that there is, first of all, the length of this bond, like where are they physically in the space, but we can also imagine that these things are, we can simplify the model so that these are fixed. But what we are interested is the angle, because depending on how these angles are organized, overall, they will tell us how does the structure look, the final structure will look like in the space, how will be this quaternary structure in the space. Okay. And there is also a funny thing that you can do, like you can make a map for a single protein of how many of these, what is the organization of these angles. There's something called the Ramachandran plot, where I have plotting here like these two axes, psi, phi and psi, which they have a specific meaning. So phi is the rotation around this bond and nitrogen bond, and the other one is the rotation around this other bond. The good news is that in TensorFlow, these people of DeepMind have implemented something that is dealing with this type of geometry. How they do more practically these things to get this angle? They take a protein, a sequence. They look for similar proteins in the uniclass data set. Also, or probably there is some kind of alignment to find out what are the similarities of this sequence. And then this sequence in this data set, they are known to have they know the structure. So because the assumption is that if you have something similar with a specific sequence, probably has a similar structure. So they look for similar things to see how they have this angle organized. And how these angles are organized, then they are mapped in some kind of you take all these similar structures, and you look how are the probability distribution of the angles in these known atoms. This is because somehow you have to build your training data set given the angles and the original sequence. And in this way, because you have to build, as we have seen, this the weights for the finance or the phase or whatever other system, we need to construct these weights in a way that they will predict, given a certain input, this conformation in space. And practically is done with in the same way. Like you need to do a gradient descent, an optimization. You are not optimizing the structure as in done in nature. You're optimizing the weights to predict the structure. So in the end, they will be, from your point of view, are just computational representations. We don't have a lot of time, but I want to show you that in the representation is they represent with a series of 2D values, which are not the images, but what they represent is that the distance between a portion of the protein and the other portion of the protein, and is this kind of representation you see here. Because this model, you know, they have a lot of like TensorFlow and all these things are highly on based on 2D images. So there was the need to represent in some kind of way in 2D what was this 3D structure. So the good news, okay, this was a lot of information. You probably have a headache now. But the idea is that given a specific sequence, if you have training this model in this way, if you have a specific sequence, you can predict a structure. And this can be applied, as I was telling you, like if you are working on finding treatments, maybe you are designing a sequence in DNA, you want to see how will appear in 3D, and then how this 3D protein will interact with something else. Let's say you're designing an antibody for corona, you can simply design it in this way, assuming that you have AlphaFold properly trained. So I don't go that much this because, and I don't expect you to. Oh, I wanted to open it. But if you have the calm and patience, it's public. So you can download the AlphaFold. It's as you can see on what is based. It's based on TensorFlow and NumPy. It's based on stuff we have already seen, plus some other things that you have to deal with data set, of course. So 3D protein structures, and you can build your own prediction of structures in this way. If I also recommend to look at this website Foldit if you want to have an idea very rough idea how to define how to design something that is interacting with these spikes in a virus or other kind of receptors. Now, I don't expect you to design a vaccine or something. I only hope that you don't see deep learning as just something to classify images. I hope now you have before and with the finance example and also with this, you can imagine and you can construct you can construct systems dealing with other type of problems. In this case was given a sequence of amino acid, building a 3D structure, a quaternary structure of a protein. Yes, I'm unmuting you, and the time is also soon. We will close it.
Yes, I have a very snappy question. The protein structure we are looking at, is it the structure, for example, like if you have the coronavirus, are we looking at the protein structure of the coronavirus or the protein structure of the human being?
You are talking of what these people are doing, or what can you do with
Yes,
AlphaFold?
yes. With what example that is cited, what these people are doing.
Okay, AlphaFold is given a sequence of amino acid, is giving a structure. It's not. It does. They don't. I mean, with this tool, they don't know what you're doing.
Okay.
These people, what they are doing, first of all, someone has to design has to came up with someone has already done this work, like for Corona, they already have. So we already know the structures, how it looks like. What we are interested is, since we know how it does look like, they have been able to understand what are made the spikes. So here I'm showing you the spike. What they are working on is to design something that interact with the spike. So we know the structure of Corona. What you can do is design something that will stop the spike to attach the human body and go somewhere else.
Oh, okay.
Because all these proteins, they are not just alone in the space. They all interact. Every protein in your body is like this. They interact each other.
Okay.
But I mean, look at the Foldit. This website
Sure.
I sent. They have a longer discussion only for Corona. But I mean,
sure.
Okay. I can show you now some results from Mohammed Al Quraishi. These are not strictly made with AlphaFold, but they give you an idea of how an original quaternary structure looks like and how a predictor structure is. So you can see that in many cases it's almost totally overlapping. In some cases, of course, different, because still this is a prediction. But it gives you an idea of of how promising these techniques are. Okay, see you.